Hello, everyone.
Hi, I had a quick question before we started class.
Sure.
I was trying to check my grades on Canvas, and it doesn't look like you've enabled us to input potential grades. So for the
Oh,
exam until
yeah,
you
yeah,
go to
yeah.
break.
Yeah, I'd be happy to do that. I'll take
Okay,
care of that after class.
thank you.
No problem there. All right. Okay. If you could type your name into chat for me, your first name or your nickname, whatever you prefer. That's my record that you came to class today. Thank you very much. Good to see everybody. Hope everybody's doing well. Okay. All right. Well, today is a lecture exam day, and I saw earlier that a lot of you have already taken the lecture exam. That's great. If you haven't had a chance yet to take it, just be sure you get it to me by 8 o'clock tonight. Today, we're going to be talking about really our last biochemical testing. Today, we'll be talking about respiration and how we test microbes for their ability to do respiration. Um, the remaining laboratory periods that we will be together, we're going to be actually talking about the coronavirus. So next week, you have your last lab practical exam. And then you'll see if you look at the schedule that we're going to spend a couple of laboratory periods talking about the coronavirus. We're going to talk about specifically the types of testing that are available to test for this virus. We'll talk about the different varieties of tests and what they're testing for. And then we'll talk about the variety of vaccines that are available and how those vaccines work, what's in them, what is being used to stimulate the immune system. So we'll spend a little bit of time towards the end of the semester talking about the pandemic virus. But today we'll be basically finishing up our discussion of all the different types of tests that we can do in the laboratory in order to identify a microbe based on its metabolic abilities, based on the kinds of things that it can and cannot do. And again, we'll be focused specifically on respiration today. So the first thing I'm going to do, as usual, is I'm just going to pull up our schedule, the modules on Canvas. We'll take a look at where we are in the course and what's coming up. So that's what you should see right now on your screen. You should see the page in Canvas after you click on modules. So I'm just going to scroll down in the modules until I get to this week, which is right here. Here's the lecture exam. And here are here's the slides for today's lab. And this is the homework questions that are associated with the lab. So this is actually the last set of homework questions that you'll be doing related to laboratory. You don't have any lectures to view this week. Hooray, you get a little break, which is great. I know you've been working really hard, but I hope you'll take the time that you might have devoted to watching lecture videos. I hope you'll use that time to get your lab notes together for next week. So next week on the 27th, you have your last lab practical exam. And since we're taking a lecture exam today, what that means is we're moving into the last set of topics in the course. We're sort of leaving behind the specifics of microbes, and instead we're looking at things like the immune system and how it responds to infection. That'll be next week. And then vaccination and some of the principles of epidemiology. And then finally, some about how pathogens create disease and how they're transmitted from person to person. So the last topics in the course, a lot of students really enjoy because it gets more into the sort of the public health aspect of microbiology. So I think you'll enjoy what's coming up. So the other thing I wanted to pull up, just for completeness sake, I'm going to go ahead and pull up our tentative schedule. So this is at the end of the syllabus. We're right here. This is the week of the 19th. It's Tuesday, so the lecture exam is happening today. Here's the lab. We won't be meeting on Thursday. Next week, the lab practical, as I said, and then we've got a couple of discussions scheduled about the coronavirus. So we're rapidly coming to the end of the semester. It'll be here before you realize it. So it's an important time to remind you to don't let up. Don't let up. I know you're tired, and I know you have a lot of other work that you're doing right now. But I also know you guys have been working hard, and I want you to keep it up through the last couple of weeks, so so you can get the good grade that you deserve. All right. Does anybody have any questions about anything? Anything to do with our schedule, or anything to do with something you might be viewing in lecture or reading in your text, or anything at all? You feel free to type it into the chat as usual. If it comes to you while we're doing the lab today, if a question comes to you, feel free to type it in, and we can talk about it. Okay. All right, I'm going to go ahead and pull up our slides. And that's what you should be seeing right now, the title slide for respiration. Let me just drop this down a little bit here. All right. So the first thing I want to do is just do a little bit of very quick review. Remember, when we use this word oxidative, when we use this term, we're not talking about oxygen. We're talking about redox reactions. So anytime we talk about oxidative metabolism, we're talking about respiration, cellular respiration, which is a long series of redox reactions, oxidation, reduction reactions. Remember, oxidation is a loss of electrons. Reduction is a gain of electrons. Microbes, many of them are capable of oxidative metabolism, as we've been discussing, but many others of them are capable of fermentative catabolism, and there are some that are able to do both. In fact, there are some that are only capable of fermentative catabolism or metabolism. So there are different ways to make ATP, cellular respiration, and what we call oxidative phosphorylation. Again, nothing to do with oxygen. This is redox phosphorylation. This is the kind of metabolism that is very, very good, very efficient at making ATP. We said that cellular respiration can be done aerobically or anaerobically, depending on the molecule that is sitting at the bottom of the electron transport chain. Fermentation is different. Fermentation will also make ATP for the microbe, but it's done differently. It's done through substrate level phosphorylation, which is very inefficient. It'll still make ATP, but it does it inefficiently. And as I said, lots of microbes are able to do both things. And there are even some microbes that are only able to ferment. They are what we call pure fermenters. So we've talked a lot about aerobic cellular respiration, respiration that uses oxygen as the final electron acceptor at the end of the electron transport chain. Remember, it's just a long, long, long series of oxidation reduction reactions. It takes this molecule glucose and it breaks it apart. It breaks all of the covalent bonds that are holding that molecule together. So the glucose gets oxidized because it's losing all of the electrons that used to create those covalent bonds. Remember, all a covalent bond is a pair of shared electrons between two atoms. When the bond gets broken, the electrons are released and they are lost to that molecule. Something else is going to receive those electrons, and that something else is going to get reduced. Now, if we look at the reduction that happens at the very end of the electron transport chain, I've got the formula right here. Now, it's not a complete formula. It's not balanced. In fact, I'm going to add this in too. But the general idea is shown in the formula that oxygen is going to get reduced at the end of the electron transport chain. All of the electrons that were released from the breakdown of glucose, all of those electrons are going to be sent down the electron transport chain, and their energy is going to be harvested. At the bottom of the electron transport chain, those electrons have to be accepted by something. Anytime something gets oxidized, something else has to get reduced. And in aerobic cellular respiration, that something that gets reduced is oxygen. It will receive those electrons. It'll also take on some hydrogen ions, and we'll get water produced. So that's what we call the pathway of reduction. That's what happens to the oxygen at the bottom of the electron transport chain in an aerobic microbe. Now, all of the electrons, as they're getting passed down the electron transport chain, the reason that they're getting sent down that chain of proteins is so their energy can be harvested. Remember, at the very end of this process, we're making ATP. So we need the energy from the glucose molecule to build the ATP molecules. Now, within the electron transport chain, which again is made out of these complexes of proteins, the electrons have to be passed down the chain, if you will, or down the stairs, as I like to think about it, from one protein to the next, to the next, to the next. So there has to be sort of a middleman molecule that helps those electrons move from one part of the chain to the next part of the chain, to the next part of the chain, all the way down. In lecture, we talked about this molecule called ubiquinone. And we also talked about this type of molecule called a cytochrome. Okay, lots of microbes have these cytochromes in their membrane, and these are molecules whose job it is to help pass electrons from one protein to the next in the electron transport chain. That's all a cytochrome is. It's like an assistant. It's helping. It's taking electrons from one of the proteins in the ETC, and it's passing them on to the next protein. So the cytochrome itself gets reduced and then oxidized. It receives electrons and then it gives them away again. Now, in bacteria, there are 4 classes of these molecules, these cytochromes. and they're referred to by letters. There's cytochrome A, there's a cytochrome B type, there's cytochrome C, and there's cytochrome D. Now I have the cytochrome C highlighted because one of the tests that we're going to look at today is going to test for this particular type of cytochrome within the microbial cell. So some bacteria have the A type, some have the B type, some have the D type, and some have this C type. They're all cytochromes. They're just different versions of cytochromes. And one of the ways that we test bacteria in order to identify them is looking for this C type. So we looked at the pathway of reduction for aerobic organisms. Now let's think about the anaerobes. Remember, we said anaerobes, the organisms that do cellular respiration anaerobically, are using a molecule other than oxygen at the bottom of the electron transport chain. You've heard me say many times that sometimes they use nitrate, sometimes they use sulfate, sometimes they use carbonate. Those are some very common inorganic molecules at the bottom of the electron transport chain. Remember, that word inorganic just means they don't contain carbon. Then there are some other molecules that get used that are organic, molecules like fumarate and triethylamine. I haven't mentioned those. Those aren't as common. In fact, the single most common final electron acceptor in anaerobic cellular respiration is nitrate. Nitrate is the one that most bacteria who are anaerobic use. So let's take a look at the pathway of reduction for nitrate. These anaerobic bacteria, they don't have oxygen at the bottom of their electron transport chain. They have this molecule called nitrate. Now, nitrate is going to get reduced. It's going to receive electrons. It's going to gain electrons that are coming down the electron transport chain. And that's going to convert the nitrate to a molecule called nitrite. So it's really important that we keep these two names separate. Nitrate is the original molecule, the final electron acceptor. And when it accepts electrons, when it gets reduced, it becomes nitrite. So we said when oxygen receives electrons, it becomes water. When nitrate receives electrons, it becomes nitrite. But here's where anaerobes are a little more complicated than aerobic organisms are. Some anaerobes will put nitrate at the bottom of the electron transport chain, and it will receive electrons and it will become nitrite. And then it's done. You're done. The process is over. But there are other microbes that don't stop there. There are other microbes that have additional enzyme systems that allow them to reduce even further. In other words, the nitrate gets reduced to nitrite, then the nitrite gets reduced. It can receive more electrons. And it's going to become a molecule called nitrous oxide. And in some microbes, it can go even further. The nitrous oxide can receive electrons. It can get reduced. And it becomes a molecule called nitrogen gas. So depending on the species of anaerobic bacteria, the final product of this reduction pathway varies. Sometimes it's nitrite, sometimes it's nitrous oxide, sometimes it goes all the way to nitrogen gas. Just depends on how far the reduction reaction continues in different microbes. So that's what I've got written out on the slide for us here. There's not one pathway of reduction here, there are three. Some microbes take the nitrate to nitrite. They reduce the nitrate and make it nitrite. Other microbes will keep going. The nitrite will get reduced to this molecule called nitrous oxide. And in other species, the nitrous oxide will then get reduced and you'll get nitrogen gas. So the pathway of reduction here is a little more complicated. It varies by species. Now I've also got a couple of pathways. This is the pathway for sulfate. When sulfate gets reduced, it produces hydrogen sulfide. When carbonate gets reduced, it becomes methane. Remember, microbes are tremendously diverse. And these anaerobic microbes, they use a variety of final electron acceptor molecules. Today, though, we're going to focus on the ones that use nitrate because it's such a common pathway. All right. So the tests that we're going to be looking at today, the first test is this test called the Oxidase Test. Now, this is the test I was talking about that has to do with that cytochrome, the specific cytochrome called cytochrome C. Cytochrome C is the molecule that's helping pass electrons down the electron transport chain. So it has to be able to receive electrons from one protein and then give those electrons away to the next protein. In other words, it has to gain electrons, that's reduction, and then lose electrons, that's oxidation. And you are probably not surprised to hear me say that there's an enzyme involved in that. And that enzyme is called cytochrome C oxidase. So cytochrome C oxidase is the enzyme that allows cytochrome C to do its work, to receive electrons from one protein and then give them away to the next protein. Now, the way this test works is very simple. It's a lot like the catalase test. It's very quick and it's very simple. Basically, you have to grow up the organism to produce some colonies on an agar plate. You're going to expose some of those colonies to what's called the oxidase reagent. If those cells in those colonies have this enzyme called cytochrome C oxidase, what's going to happen is they're going to turn that reagent into this molecule called indophenol blue. Now, what's important to know about this reaction is that it's going to turn a color. Indophenol blue is going to produce this purple blue color. Now, here's something else to know. When a particular microbe is positive on the oxidase test, and remember that means it has cytochrome C oxidase, it also tells us a couple of other things, right? It tells us it must use cytochrome C in its electron transport chain. And that means it also must use oxygen as the final electron acceptor. So the primary interpretation of the oxidase test is that you have a microbe that it has cytochrome C oxidase present in its electron transport chain. But we can also deduce from that it's using the C type of cytochromes and that it's using oxygen as its final electron acceptor molecule. Now, here's something important to put in your notes. Put a little star next to this. If an organism is negative on the oxidase test, if it comes up negative, if there's no color change to blue, purple, that does not mean that it doesn't use oxygen at the end of its electron transport chain. We can't say that. And that's because it may be using a different family. It may be using cytochrome A oxidase or cytochrome D oxidase. It's just not using cytochrome C oxidase. So it's important that you don't read a negative oxidase test as meaning, oh, this must be an anaerobe. This organism must not use oxygen as its final electron acceptor. We can't make that conclusion. All we can say is it doesn't use cytochrome C oxidase. Okay? So that's a strange distinction, I know, but it's an important one because it's tempting sometimes when we see a negative result to interpret it as the opposite. If we say, oh, a positive means cytochrome C oxidase, and it means oxygen is at the bottom of the electron transport chain, then a negative must mean no oxygen at the bottom of the electron transport chain. But you can't say that. All you can say is a negative oxidase test means that that organism does not have cytochrome C oxidase in its electron transport chain. All right. So this oxidase test is actually really straightforward, as I said a minute ago. Here, I'm just skipping forward a little bit. What we do is we take the organism that we're testing and we grow it up on an agar plate, any kind of nutrient agar plate, basal medium, whatever kind of general culture medium you want to use. You just need to grow up some colonies, because what you're going to do is you're going to take one of these sterile swabs, cotton swabs, and you're just going to scoop up 1 or 2 colonies off of the plate. That's plenty of cells. It is possible to do an oxidase test with broth, but it's much easier to read the result when you use colony material because you're going to have lots and lots of cells. Okay, so usually we use colonies grown up on an agar plate. We just pick up 1 or 2 of them on a cotton swab, and then you're going to take the oxidase reagent and you're going to drip it down onto those cells. You just drip it right onto the cotton swab, and that's all you have to do. If it's positive, it's going to turn this purpley blue color. If it's negative, it won't. Very, very basic test and very easy to read because it's quick. Now, a couple other things here. You can see that down here, what they did was they took the oxidase reagent and they just dripped it right onto the plate, right on top of the colonies themselves. In other words, they didn't pick up the colonies with the swab. They just left them on the plate and dropped the oxidase reagent right on top. You can do that. That's fine. You can see it turned purple because cytochrome C oxidase was present. Most of the time, though, people don't do that because if they've grown up a nice agar plate like this, they may want to use these colonies for something else. So they don't want to contaminate the plate. So most of the time, we're going to pick up a few colonies and put the reagent right onto the swab. Now, it's important to note, here's another thing to know about the oxidase test. You don't want to use your metal loop. We don't use our metal loops for the catalase test. Remember, the catalase test is the one that tests for the catalase enzyme. That was the one with hydrogen peroxide, the one that bubbles. You don't want to use your metal loop for that test because it can interfere with the reaction. And the same thing is true in the oxidase test. So that's why we use the swab instead. You can use a cotton swab to pick up some colonies, or you can use one of the plastic loops, the disposable loops. That would be fine too. But you want to avoid the metal loop because you can mess up the results with a metal loop. Here's something else to know about the test. The reagent comes in a couple of different forms. You can buy a little bottle, a little squirt bottle with the reagent in it. And when you open the bottle for the first time, the reagent starts to degrade. In other words, once the bottle is opened, it has a limited shelf life. Generally just a couple of weeks, believe it or not. So that means if you're in a lab that does a whole lot of oxidase testing and you're going to run through the reagent very quickly, it's no big deal. But if you're in a lab that doesn't do a lot of oxidase testing, you're probably not going to get to finish all of the reagent in the bottle before you have to throw it out. Because once you open it, in other words, once it gets exposed to the air, it starts degrading. It has a very limited shelf life. So there's a way around this. In labs that don't do as much oxidase testing, instead of buying the bottle of liquid reagent, they buy these little test strips instead. These are just pieces of filter paper, basically, that have been soaked in the oxidase reagent, and then they're packaged. They're packaged individually. So you open up one strip at a time or two strips at a time. And that way they last much, much longer. If you're in a lab that's not doing that much oxidase testing, it makes sense to use the strips. Because once you open that bottle of liquid reagent, it's got a very limited shelf life. So if we go back to the slides, on the upper right-hand side here, you can see what these strips look like. They're just little strips of paper, literally. And they've been soaked in the reagent. So all you do is you scoop up one or two colonies again with your cotton swab. And all you have to do with your swab is just rub it onto the strip. If the cells are oxidase positive, it's going to turn purple where you rub it. If it doesn't change color, it's a negative. So very, very easy to use these strips. And because they're individually packaged, they last a long time. Much more practical. All right. Now, in terms of examining the reduction of nitrate, remember, examining whether a microbe is using nitrate at the bottom of its electron transport chain, we have this test referred to as the nitrate reduction test. Now, this is one of the most complex tests we've talked about this semester, which is part of the reason we save it for last. This test has several steps to it. So you have to know not only what the steps are to the test, but you also need to know how to interpret each step. So basically what this test is doing is testing for the ability of this microbe to reduce nitrate to nitrite. So that was the first pathway that we talked about for nitrate being reduced. If the microbe can take nitrate and use it at the bottom of its electron transport chain and convert it, in other words, reduce it to nitrite. That's what the test is all about. Basically, this is just a plain old nutrient broth that's been supplemented with nitrate. We just put lots of nitrate into the broth. So you're going to take your cells, whatever kind of microbe you're testing, and you're going to inoculate the broth. And then you're going to incubate the tube for about 48 hours. We let these tubes incubate longer than normal because we want to make sure that the microbe has plenty of time to reduce the nitrate in the tube. Now, here's where it starts to get complicated. When you take the tube out of the incubator, it's going to look like nothing has happened because nitrate reduction test broth is just beige. It's very boring looking. When you put it in the incubator, it's going to be beige. When you take it out of the incubator, it's going to be beige. So when you take it out of the incubator, you have to add two reagents to it. What the reagents are going to do is basically test for the presence of nitrate. If there's any nitrate in the tube, when you add these two reagents, the tube is going to turn red. So remember, we said the first pathway for the reduction of nitrate is that nitrate becomes nitrite. So if the microbe that's growing in that tube has used the nitrate as its final electron acceptor, after 48 hours, there's going to be lots of nitrite in that tube. It has taken the nitrate and it has reduced it to nitrite. When we put the two reagents in, the nitrite in the tube will react and the tube will turn red. That is a positive test result because we've just shown that that microbe is able to take nitrate and reduce it to nitrite. It's using the nitrate at the bottom of its electron transport chain. Pretty straightforward so far, right? All right, let's go back to the slides. Now, these two reagents have long, complicated names. We don't need to know the names of the reagents. Everybody refers to them as either reagent A and reagent B. And in some test kits, they're called reagent 1 and reagent 2. So it doesn't matter that you know their actual molecular names, chemical names, just that you know that there are two different reagents that have to get added to the tube. And if there's nitrite in the tube's going to turn red, and that's a positive test. Now, if you put the two reagents in and the tube does not turn red, you might think, oh, well, this is a negative result. Here's where it gets complicated. It's not necessarily negative. It could be negative, right? Maybe this is an organism that doesn't reduce nitrate. Maybe what's sitting in the tube right now is just nitrate because the organism couldn't reduce it to nitrite. If that's the case, then yeah, that would be a negative result. But remember, some microbes can not only reduce nitrate to nitrite, but they can then take the nitrite and reduce it even further. So maybe the reason there's no nitrite in the tube is because it's been further reduced. That would be a positive result. So when we take the tube out of the incubator and we put reagents A and B into the tube and it stays colorless or beige, it could be positive or it could be negative. We don't know. So what are we going to do? How are we going to figure that out? Well, the next step is to add another reagent. So if there's no red color after you put reagents A and B in the tube, you're going to add the next reagent, which is zinc. It's actually a zinc metal. It's a powder formulation of zinc. Now, if there is any nitrate in the tube and you add zinc, the zinc will reduce it to nitrite. And guess what's going to happen? It's going to turn red. Remember, you already put A and B in step 1. So A and B is still in there. We're going to add zinc. If there's any nitrate in that tube, the zinc is going to turn it to nitrite. It's going to reduce it to nitrite, and you're going to get a red color. But here's the kicker. That's a negative result, isn't it? That's a negative result. The microbe didn't reduce the nitrate, right? The microbe didn't turn the nitrate to nitrite. The zinc did. That's a negative test because that tells us that there was nitrate left in the tube, which means the microbe couldn't reduce it. There must have been nitrate in there because when we put the zinc in, the zinc will reduce any nitrate to nitrite. So if you get a red color after you put zinc in, you've got a negative result for nitrate reduction. Now, what about the other possibility that we mentioned? What about the possibility that this microbe reduced nitrate to nitrite, but then took the nitrite and reduced it further? How do we know if that's happened? Well, if we put zinc in and it's still colorless, it's still beige, now we know that the bacteria must have further reduced the nitrate to one of the two products I mentioned earlier, either to nitrous oxide or to nitrogen gas. Now, how do we know? How do we know which pathway it used? Well, because the nitrous oxide will dissolve into the liquid and the nitrogen gas won't. So we're going to put our old friend into this tube. We're going to put a Durham tube into the nitrate reduction test. We're going to put one of those little tiny upside down glass test tubes into this test. And that way, if after we've added reagent A and B and after we've added zinc, the tube is still colorless, we can look in the Durham tube. If there's gas in the Durham tube, we know that the nitrate was reduced all the way to nitrogen gas. If there's no gas in the Durham tube, we know that the nitrate was reduced to nitrous oxide, and the nitrous oxide dissolved into the medium. So you can see it's a very complex test. And the reason it's complex is because just because a microbe uses nitrate at the bottom of its electron transport chain, it's not nearly as simple as oxygen. When oxygen gets reduced, it just makes water. Done. But when nitrate gets reduced, it can go to nitrite, which can then get reduced to nitrous oxide, which can then get reduced to nitrogen gas. There's 3 different products, in other words, that can be made. And which product you're going to get depends on the species. The only difference is whether or not that organism has the enzymes it needs to make nitrogen gas or to make nitrous oxide or to just stop at plain old nitrite. So it's the fact that there are so many end products here that makes this test so confusing. All right. So we take some cells and we inoculate the nitrate reduction test broth, which is just a nutrient broth that we have supplemented with lots of nitrate. We also put a Durham tube in there, an upside down Durham tube. Now we're going to put that tube in the incubator for 48 long hours because we want to give it plenty of time to reduce as far as it can reduce. When we take the tube out of the incubator, we're going to add reagents A and B, and we're going to look for a color change. These reagents, in the presence of nitrite, are going to turn that tube red. So if the microbe used the nitrate in the test medium and reduced it, added electrons to it, it's going to become nitrite. Reagents A and B will react with the nitrite, and you're going to get a red color. That's a positive result, because that tells you that the microbe must have reduced the nitrate to nitrite. And those two reagents showed us the nitrite. Now, if we put A and B in and we get no red color, if the tube stays that colorless beige color, what do we do then? Well, we have two possible explanations. Either the nitrate is still in the tube because the microbe could not reduce it, or the microbe reduced the nitrate to some product other than nitrite. It either turned it into nitrous oxide or all the way to nitrogen gas. Now, how do we know which explanation is correct? We add zinc. We have to add zinc to distinguish between these two. So we add the zinc reagent, this metal powder, into the tube. If the nitrate is still in the tube, that was explanation number one, the zinc metal will reduce it into nitrite, and the tube will turn red. But that's a negative test result. And the reason it's negative is because the microbe didn't reduce the nitrate. The microbe couldn't. That's why the tube was still full of nitrate. The microbe couldn't do it. The zinc did it. The zinc turned the nitrate into nitrite, and the tube turned red. So that's a negative test result. If you add the zinc in and the tube still doesn't turn red, now you know your second explanation must be correct. That the microbe reduced the nitrate, but it went further than nitrite. It produced another product. It either reduced the nitrite to nitrous oxide, and that nitrous oxide, again, will dissolve into the liquid, or it took the nitrite, reduced it to nitrous oxide, and then reduced that to nitrogen gas. Either way, though, that's a positive test because reduction was going on here. Nitrate got reduced. It's just that it got reduced to one of these alternative products. How do we know which product? Well, we look for the gas. We look for the gas in the Durham tube. If there's a bubble in the Durham tube, we know it's nitrogen gas. If there's no bubble in the Durham tube, we know the final product was nitrous oxide. Make sense? Does any of it not make sense? This is an unusual final electron acceptor molecule, this molecule called nitrate. It can get reduced quite extensively, can't it? It can get reduced to nitrite, but then nitrite can get reduced, and you'll get nitrous oxide. And you can even reduce nitrous oxide if you have the right enzymes. And now you've got nitrogen gas. At that point, we're done. You can't reduce the nitrogen gas any further. But depending on the species of microbe, depending on the type of bacterium, sometimes the reduction of nitrate produces nitrite. Sometimes it produces nitrous oxide. Sometimes it produces nitrogen gas. And by using this test, we can know which. We can know what pathway that microbe is using. So it's a really very helpful test for these anaerobic cellular respiration organisms. All right. So let's do a couple of these tests using some organisms. We're going to use 2 gram-positive cocci today, and we're going to use 2 gram-negative rods. We're going to use an organism called Streptococcus pneumoniae. We're going to use one called Staphylococcus epidermidis that you've seen before. We're going to use our old friend E. coli and this organism called Pseudomonas aeruginosa. Remember, all of these organisms can be found in the human body and not necessarily be pathogens. But there are strains of these organisms that can be pathogenic. Streptococcus pneumoniae and Pseudomonas aeruginosa especially can have some pathogenic strains that can cause disease. All right. I'm going to pull this up first. So remember, we said we have the oxidase test that we're going to look at today, and we have the nitrate reduction test that we're going to look at today. So here on this slide, you're looking at the 4 organisms and their oxidase results. So I took each one of these organisms separately and I streaked them out onto an agar plate, just a plain old nutrient agar plate. And I grew up individual colonies of each one. I did a quadrant streak technique. Remember, that's the kind of streaking that we do when we want to be sure to get individual colonies forming. I took a few of the strep pneumonia cells onto a cotton swab and I dripped the oxidase reagent on them, and I got no color change. I picked up a few of the staph epidermidis cells, no color change. E. coli, same thing, negative oxidase test. But the Pseudomonas produced a positive result. When I put Pseudomonas onto the cotton swab and I dripped the reagent on, it turned this purpley blue color. So that's an interesting fact about the Pseudomonas organism. Pseudomonas aeruginosa is oxidase positive. Remember what that means. That means that this organism uses the type C cytochrome in its electron transport chain. How do I know? Because what this test is testing for is the enzyme called cytochrome C oxidase. It's using that enzyme to help cytochrome C do its job, which is passing electrons from one protein to the next down the electron transport chain. I also know, as a result of this test, that Pseudomonas aeruginosa must be doing aerobic cellular respiration, at least as one of the pathways it's capable of. In other words, Pseudomonas aeruginosa must use oxygen at the bottom of its electron transport chain. Now, here's that point I made earlier. E. coli was negative for oxidase. Staph epidermidis was negative. Strep pneumonia was negative. Does that mean that these don't use oxygen at the bottom of their electron transport chain? Can I say that? No, I can't. I can't say that because they may be using a different family of cytochrome. This test only tests for the cytochrome C oxidase. So it is possible, in fact, we know that E. coli, for example, is a facultative anaerobe. It can use oxygen, yeah, but it must be using a different cytochrome family. That's all you can say, okay? So you have to be careful how you interpret your negative oxidase tests. All it means, if it's negative, is that that organism does not have cytochrome C oxidase in its electron transport chain. All right. Now, the nitrate reduction test. I put this in here. I like this image. This is a flow chart for the nitrate reduction test. I hope you find this as helpful as I do. Remember, we inoculate this nitrate reduction broth, which is just a nutrient broth that has been supplemented with nitrate. We inoculate it with each of the organisms. We incubate it for 2 whole days, and then we read it. Take it out of the incubator and add reagent A and reagent B. These are the names of each one of those reagents, but don't worry about that. You don't need to know those names. When you add those reagents, one of 2 things is going to happen. Either the tube is going to turn red or the tube is not going to turn red. That's the only 2 things that can happen. This is what the tube looks like when it stays beige, right here. This is what the tube looks like when it turns red. It's a very obvious color change. So if I add reagent A and reagent B and it turns red, I know my microbe is positive for the reduction of nitrate. It is able to reduce nitrate to nitrite. If the tube stays colorless after I add reagent A and B, I'm not done. I got to keep going. I don't know what's happening yet. Okay. Could it be negative? Yeah, sure it could, but it might not be. It might also be positive. Okay. I have to add the zinc dust to know for sure. When I add the zinc, if it turns red, that's a negative test. The microbe was not able to reduce the nitrate. The zinc did it. The zinc took the nitrate that was still in that tube after 48 hours and it reduced it to nitrite. That's what made the tube red. If you add the zinc and the tube stays colorless, that is a positive result for nitrate reduction. It's just that the nitrate got reduced to either nitrous oxide or nitrogen gas. And the way I know which one happened is by looking in the Durham tube. If there's gas in the Durham tube, I know that the microbe reduced the nitrate to nitrogen gas. If I see no gas in the Durham tube, then I know that the microbe reduced the nitrate to nitrous oxide, and the nitrous oxide dissolved into the medium. So here's our results. Streptococcus pneumoniae is over here on the left. Staph epidermidis is in the middle. Here's E. coli. Here's the Pseudomonas. Now you'll notice there's some text here because it's telling us where these colors came from. Look at Staphylococcus epidermidis and E. coli first, because they have the same color here, don't they? Both of these tubes turned red after reagents A and B were added. Now here they're calling them reagents 1 and 2, but that's the same thing. So we took the tubes out of the incubator, we added A and B to them, they turned red. What do we know now about those two organisms? Are they nitrate reduction positive? Or are they nitrate reduction negative? Or do I need to do something else?
They're nitrate reduction positive.
They're positive. Absolutely. We're done with them, right? We're done. Tube turned red after reagents A and B were added. That's a positive test. What that tells us is that the nitrate that was in the medium, in the culture medium, is all gone. It got reduced to nitrite because that's what A and B react with. They react with nitrite. So there must have been nitrite in the tube. And the only way the nitrite got there is if the microbe reduced the nitrate. So that's a positive result. Now, it gets a little trickier with the other two. The Streptococcus pneumoniae tube stayed colorless after reagents A and B were added. So we put some zinc in, that's the ZN, and it turned red. Nitrate positive, nitrate reduction negative. Do I need to do something else?
Negative.
Negative. Good. Negative. There must not have been any nitrite in the tube because if I put A and B in and it stays colorless, that tells me there's no nitrite in there. But I don't know yet what happened. I got to do the zinc part. I add the zinc in and it turns red. Remember, zinc will take any nitrate that's present and reduce it to nitrite. So if you get red after you add zinc, it's a negative test because the microbe didn't reduce the nitrate, the zinc did. Okay, and finally is our friend Pseudomonas. Pseudomonas just stayed colorless. After we added A and B, it was colorless. After we added zinc, still colorless. So we know that this is a positive result. Pseudomonas aeruginosa is capable of reducing nitrate. It's just that it didn't stop at nitrite. It reduced the nitrate even further. The question now is, did it reduce it to nitrous oxide or did it reduce it to nitrogen gas? And we have to go to the Durham tube for that. I know it's really hard to see, but the Durham tube is right here. And you can just see this little white line right here. That's the liquid interface there. This is a big gas bubble right here. So Pseudomonas aeruginosa is positive for the reduction of nitrate. And it reduced it all the way to nitrogen gas. And I know that because there's gas in my Durham tube. Positive result. Now, I almost hate to mention this, but I'm going to. It should make sense once you've reviewed. If I take a nitrate reduction test out of the incubator after 48 hours of incubation, before I do anything to the tube, if I look in the Durham tube and I see a gas bubble in there, I know I've got a positive result. If I take my tube out of the incubator after 48 hours, before I do anything else, before I put A and B in, before I put the zinc in, before I do anything, I just look at the Durham tube and I see a gas bubble in there, I have a positive result. Does that make sense? I have a positive result because what that means is there's nitrogen gas in the Durham tube. And the only way the nitrogen gas could be there is if the nitrate was reduced. So it's a little bit of a cheat to take a tube out and look for the gas bubble. And of course, if there's no gas bubble, I have to do the rest of the test, don't I? If there's no gas bubble, I cannot say, oh, it's negative. I can't say that. I have to go through the steps. But it is a little extra sort of a trick on this test that if you take the tube straight out of the incubator and you already see a gas bubble, you know you have a positive nitrate reduction test. And you know that the microbe reduced the nitrate all the way to nitrogen gas. Just a little trick on that test. All right. So just another couple of words about this organism, this Pseudomonas organism. It's an interesting organism. It comes up oxidase positive, which, as you can see, at least among this group of organisms, is a little bit different. There are lots and lots of bacteria that are oxidase positive, but Pseudomonas is one of them that is kind of an interesting organism. It also happens to be nitrate positive, and it reduces nitrate all the way to nitrogen gas. Pseudomonas is an organism that can live in and on the human body and not cause us any trouble at all. Unlike many other types of bacteria, it can also live out in the environment, though. This is one of the bacteria that is perfectly happy to live off of our body. And it happens to be one of the bacteria that loves to be in dirty, wet, dark places. So anytime that you have a facility, for example, that isn't kept quite as clean as it should be kept, Pseudomonas is a concern, because Pseudomonas loves to grow in dark, wet corners, basically. You find it in drains all the time, in sinks, on the bottom of faucets. You know, when sinks stay wet all the time because people are using them all day? Pseudomonas loves to grow there. And like I said, a lot of strains of Pseudomonas are perfectly harmless, but the ones that are pathogenic are a problem for us in healthcare. Pseudomonas is an organism that is called an opportunistic pathogen. And what that means is, in a healthy person, a person who's otherwise perfectly healthy, this pathogen may not have an opportunity to even cause disease because the immune system is going to get it. But if you're a person who is suffering with some other illness, let's say you're in the hospital because you just had a massive heart attack, or maybe you have pneumonia from another organism. Maybe you're a burn patient and you've lost some portion of your skin to a burn. Pseudomonas is the kind of pathogen that loves to get into those patients and cause disease. It's opportunistic. That means it takes advantage of the opportunity of the immune system being busy, the immune system being distracted by some other disease. So anytime you get a patient into a hospital who is compromised, who is very ill with something else, they run the risk of picking up an opportunistic infection like Pseudomonas. It's especially problematic in, like I said, burn patients. Pseudomonas can cause terrible, terrible infections in burn patients. You'll find it in patients who have to have long-term catheters placed. Think about, for example, patients who are in long-term care facilities and who have to have a catheter, a urinary catheter placed because they can't get up and down and urinate by themselves. These people are very vulnerable to picking up these opportunistic infections. So something to know about Pseudomonas. I could tell you one other thing about Pseudomonas because it's also a problem in veterinary medicine. It's also an opportunistic pathogen in animals. When you get a Pseudomonas infection, you will know it as soon as you smell it. Pseudomonas has a terrible odor to it when it's growing. So yeah, I have dealt with it in animal patients on more than one occasion. All right, does anybody have any questions about either the oxidase test or the nitrate reduction test? Any questions about those two? It's a good idea in your notes to maybe copy down that flow chart for the nitrate reduction test. You know, if this happens, then do this. And if this happens, then it means this. Yeah, it's a tough test. You just have to keep in mind all the steps and what each step means in terms of interpretation. All right. If there's no questions, I'm going to let you go. Remember, if you haven't taken the lecture exam yet, be sure to get that to me by tonight. There's no lectures to view this week. You do have lab homework to do, but there's no lectures to view, so there's no lecture quizzes. So this is a good week to get your lab notes in order. If you had to miss a lab, maybe, you can go onto our playlist and watch it and make sure your lab notes are in really good order. And then you'll be all set for the lab practical exam next week.
I did have a question about the final exam. Is
Yeah.
it cumulative?
The final is not cumulative in here. The
Okay.
final is essentially the last lecture exam.
Okay, perfect. Thank you.
You're welcome. All right, everybody, I'm going to let you go. Have a good evening. Don't hesitate to get up with me if you have any questions that come up. And I will talk to you next week.
Thank you. Have a nice night.
All right, you too.